Hello and welcome. The American Society of Brewing Chemists and the Brewers Association have teamed up to bring you this collaborative instructional video series on basic brewing microbiology. My name is Eric Jorgensen with the Highland Brewing Company, and our topic today is beer spoiler screening with HLP media. Beer is a pretty microbiologically resilient beverage. No human pathogens can survive in beer, which is partly why its consumption has been so widespread throughout human history. However, a few bacterial strains can spoil the flavor and appearance of a beer by turning it sour and turbid. The bacterial species Lactobacillus and Pediococcus are the most common beer spoilers encountered in today's craft breweries. Brewing microbiologists have developed many types of growth media to screen for them. Today we'll be talking about one of the more common medias used in the United States for this purpose, called HLP. HLP stands for Shoes, Lactobacillus, and Pediococcus media. This is a type of selective media, which means that it selects for the bacterial species Lactobacillus and Pediococcus and selects against all others. So here's how it works. HLP contains sugars, vitamins, trace elements, proteins, and the building blocks for amino acid production necessary to promote growth of lactic acid bacteria. Malt extract, yeast extract, tomato juice broth, sodium sulfate for cysteine and methionine production, and vitamin B5 fulfill these nutritional requirements. In addition, several active ingredients select for organisms of interest. Citric acid is added to adjust the pH down to optimal growth conditions for lactic acid bacteria. Mercaptoacetic acid, whose conjugate base is sodium thioglycolate in this media, is used to scavenge any dissolved oxygen and creates an anaerobic condition inside the HLP tube. This means that you don't need an anaerobic incubator, which is partly why this media is so popular with craft breweries. Agar gel is a thickening agent used to keep the sodium thioglycolate evenly distributed throughout the tube. Otherwise, you would have an oxygen gradient throughout the tube with aerobic conditions near the surface and anaerobic conditions near the bottom. Sodium acetate suppresses the growth of gram-negative bacteria. Cyclohexamide, also known as actidione, inhibits protein synthesis in eukaryotes. It has no effect on prokaryotes. This means that yeast growth is suppressed while bacterial growth is allowed to flourish, making it an ideal compound to screen for bacteria in breweries. Now be warned, you are also a eukaryote. Cyclohexamide is a potent toxin, so consult your material safety data sheets and follow safety procedures. So now we've got this great cocktail of nutrients for microbial growth, but we've selected against yeast, gram-negative bacteria, and aerobic microbes. The only category of microbes that will thrive in HLP are gram-positive, anaerobic bacteria. In brewery settings, that is usually Lactobacillus or Pediococcus. So here's how to use it. Simply boil 7 grams of HLP per 100 milliliters of reagent water, which means distilled or deionized water. Boil it for 2 to 3 minutes and transfer to some sterile culture tubes, and you're ready to inoculate with your sample. One good thing about HLP is that you don't need to autoclave it. This makes it a very easy media to use when you're just starting a micro program. Use proper sterile technique. Remember to work around your flame, keep your gloves and tools sterile, and be careful of anything that is above and around your samples. Once inoculated, incubate your culture tubes for at least 3 to 5 days. After incubation, you want your media to be nice and clear. Colony growth could indicate a beer spoiler contamination. You may find a little haze in yeasty beer samples, such as those taken from early fermentation. Lactobacillus will typically form little swirly colonies in the media, while Pediococcus will typically form little balls. But it is important to note that this is not always the case. In badly contaminated samples, often you cannot distinguish individual colonies and will see only opaque growth. Every now and again, you may even find a colony of cyclohexamide-resistant wild yeast. It is important not to simply look at the colony morphology and assume you know what organism you are dealing with. Find out. Validate or invalidate your assumptions by extracting a sample of the growth, gram staining it, and observing its cell morphology under the microscope. At Highland, we use 15 milliliter reusable glass culture tubes with an airtight closure. We inoculate 10 milliliters of HLP media per tube with 1 milliliter of beer sample. We mix the tube and we incubate for 5 to 7 days. After interpreting the tubes, we clean, sterilize, and reuse them. Now, with this setup, we have a detection limit of 1 colony-forming unit per milliliter, so it's one of our less sensitive tests, but it'll catch bad problems, and it's an important media to have on hand if we want to verify colony identification from one of our more sensitive tests. More information on different types of selective media is available to ASBC members on their website. Thanks for watching.